the fundamentals of energy harvesting by living organisms. And the initial thing always comes on Earth from the light from the sun, and that happens in plants. The photons are absorbed in the chloroplast, that is an organelle within the leaves, and that is with these inner membranes inside. There are membrane protein complexes that are absorbing the photons, and as a result, that causes electron transfer reactions that come through these various, and there's two light absorbing protein complexes, and it causes the reduction of NADP to NADPH, which then is an energy source for living things. And it also causes protons to be pumped to the inside of these little vesicles, the thylakoids. Ultimately, those protons are then used to power the ATP synthase. It's called F1 for this complex and FO in the membrane to convert ADP and phosphate into ATP. The same thing occurs in mitochondria, another energy harvesting organelle, by the process of respiration, where you're taking the reduced, in this case, NADH, and oxidizing it, and in so doing, you're moving electrons through these protein complexes that are embedded in the membrane that then serve as proton pumps to the inner part of the inner membrane of the mitochondria. And again, this non-equilibrium proton concentration across the membrane is used as an energy source to power the synthesis of ATP by the F1-FO-ATP synthase. So I'm going to talk now about how the F1-FO-ATP synthase works, what we have learned, mainly through the development of novel single molecule technologies. So here is a brief video of cartoon video of how the F1-FO works. In fact, it's a molecular motor. Here's the non-equilibrium proton concentration difference across the membrane here, and that energy is known as the proton motive force. So then, as the protons want to move back toward equilibrium to equalize the proton concentration across the membrane, there is a channel through the FO that is much more efficient than going across the membrane by itself. And in so doing, it uses that energy to generate torque to rotate the rotor here in FO. And that's attached to the drive shaft in F1, and that rotates inside and converts changes the conformations of these protein subunits to convert the ADP and phosphate to ATP. So in fact, what it's doing is driving the synthesis of ATP away from equilibrium. So it's there in much higher concentrations than it would be at equilibrium. And that's really what, then it's that disequilibrium, chemical disequilibrium, that gives rise to the energy that powers most of life's processes, which in animals includes muscle contraction and mental activity. So the various organisms can drive this concentration difference away from equilibrium to different degrees. So for example, the bacteria E. coli can drive it to about 0.1 on the log scale, whereas a cat, when it's just lying around sleeping during the day, can generate twice that amount of energy. But of course, after exercise, and it consumes a lot of the ATP, you can see how that is depleted by this process. And of course, when the cat dies, then it goes back to equilibrium, and that's the equilibrium position. So this is the difference that in the energy that's available to the organism. So this works because this ring called the C ring rotates relative to this peripheral stalk that contains subunit A, that contains the channels for the protons to go through. And then this gamma subunit that protrudes down in here, that's also part of the rotor, that's the drive shaft. Now, we can pop the F1 portion off of the membrane and away from FO, and we can study that separately. Under these circumstances, then when you add ATP, you can drive the reaction backwards. It hydrolyzes the ATP, and it'll drive rotation in the opposite direction. So an ATP will come into one of the 3 catalytic sites in the ring that drives rotation by 120 degrees, and product, ADP and phosphate, will come off from a different site. Here comes the second ATP onto that empty site, and then it drives a second 120-degree rotation. So this is called the alternating site mechanism, in which each of the 3 catalytic sites is operating at the same time, but out of sync with each other, so that each one is driving ultimately the entire rotation of the process. Now the movie starts to work. Okay. All right. So the first crystal structures showed that for the F1, I'm going to start by explaining how the F1 works and what we've learned about it. That the first crystal structures showed that in the static structures, you would have the alpha and beta subunits that are in alternating going around the central core of the gamma subunit. And at any one time, you're going to have one empty site, one site where you have an ADP bound, and one site where you have an ATP bound. The binding sites for the catalytic sites are in between the alpha and beta subunits, but it's mostly the beta subunits that are doing the work. They have most of the amino acids that form the catalytic process. So if we just look at a cross section from the side of this, you can see here's the gamma subunit, and there are 2 domains. There's this coiled coil domain that protrudes down the drive shaft, and then there's also the foot domain that protrudes out from the base, and that connects up here to the C ring. The beta subunits have 3 domains. There's this barrel domain, and both alpha and beta subunits have these 3 domains. And the barrel domain connects up like a crown, and it forms a rigid structure at the bottom, so that all the other movements are relative to that barrel to allow torque to be generated. Then this green domain is where ATP binds and is hydrolyzed or synthesized during ATP synthesis. And finally, there is this lever domain. So while the phosphates of the ATP are mainly binding down here, the base of the nucleotide is binding up here. And so you can see where you have it's empty, this lever arm is open, and where you have a nucleotide bound, in this case ADP, here it is right here, that pulls the lever arm into the closed position, where now it's tucked under the foot domain. So that's an important part to understand about the structure, and now we'll see how that relates. Okay, so a big step forward was made in understanding how it really works. But you have a crystal structure, you can see the detail, but you can't see it work. You're stuck with a static structure. So this single molecule experiment was devised where you took the F1 that's purified away from the membrane, You modified here, putting his tag so that now it would stick to a microscope cover slip. You modified this with the gamma subunit with a biotin, so now it would bind very tightly to streptavidin or vitamin B that is then attached to a long actin filament that is decorated with fluorophores, so you can actually see it under the microscope. This is only about 12 angstroms in diameter, so it's much, 12 nanometers in diameter, so it's much too small to see. But with this long, that actin filament that's several thousand nanometers in length, you can see it. And now when you add ATP, you can see that it rotates in the counterclockwise direction. So a lot of people, when I ask, do you know of this work? They say, well, I didn't read the paper, but I saw the movie. You can say that now too. Anyway, while this was, really striking in its day, and it does show clearly that it's rotating and rotating in the counterclockwise direction, the actin filament is so long and provides so much drag on the surface that it's going much slower. What you're seeing is a much slower rotation than it would be if it were operating without any drag and just being the rate limited by the internal workings of the motor by itself. So in order to understand how the motor really works, you want to eliminate this drag, and you want to devise a system that can actually capture the rotation at its real speed. This, of course, is a standard VCR video at 30 frames a second, it was really an amazing accomplishment in its day. So we devised a different method that would enable us to look at much higher resolution in much shorter time frames than what was previously possible. And what we did was to attach a gold nanorod that would be about 35 nanometers in width by about 75 nanometers in length to the gamma subunit by the same biotin streptavidin as was done previously. But these are very small, and so they don't, by a number of experiments, we determined that they don't provide sufficient drag to slow the motor down. So the unique way that we're seeing the rotation is not by actually watching it rotate in several frames, but by the fact that these nanorods have a special light scattering property. They scatter green light across the short axis and then red light across the long axis. So as it rotates, it'll flash red and green. So here's a field of view under the microscope, and you can see the blinking ones, and they blink red and green as it rotates. Each one is attached to an F1 molecule that's rotating. In the prior example with the actin filament, you're lucky if you see 3% of the molecules rotate, because the actin filament gets stuck on the surface. It's just not very good. Here we're seeing about 70% of them rotate. And so what we do now is we hook this up to our detector that we devised, and we take a nice one that's nicely blinking. We line that up with a pinhole in the microscope, and we feed the light so that it only captures the light from that one nanorod, goes through a red filter that will only allow the red to proceed, not the green, then a lens that focuses it onto the single photon counter, and it goes through a polarizing lens. That's because we realized that the light being scattered off the nanorod is polarized. And so consequently, what we see is that the strongest intensity of light is under this condition for the green light when it's exactly perpendicular, and the strongest for the red when it's exactly parallel to the long axis. The single photon counter can capture and count single photons from the nanorod and it can do so at very, very high frame rates. So here we started out, and just as an example to test the system, we sample the data at 1 kilohertz, that's 1,000 frames a second, as opposed to 30 frames a second. And so we'll collect for a few seconds. And now because we have the filter involved, this appears black when the polarizer is perpendicular. And so we collect for a while and we see very low intensity down here of light. And then we rotate, collect, and what happens is you'll see that as we monitor the rotational position, we see a sinusoidal change as a function of the rotational position. It's maximum at 90 and close to zero intensity at 0. And the nice thing about the nanorods is that they're not fluorescent, so they don't photobleach like the fluorophores attached to the actin filament. So with the fluorophores, you only really get a few minutes to collect your data before everything turns black, but this just doesn't happen. So we can collect thousands and thousands of these rotational events. So when we do that and we look at what is the error from the distribution here that we get at any different angular, you can see that our ability to measure the accuracy of our rotational position is really quite good between 0 and 90. And of course, the error gets worse as the intensity goes up. It's a function of the number of photons that we're getting in there. Okay, so now what happens when we put the F1, attach the nanorod to the F1, like down here? So now when we repeat that experiment, now it goes 120 degrees and then it pauses for a catalytic dwell where ATP is hydrolyzed, then resumes for the next 120 degrees and pauses again. And so the time that it's actually doing the power stroke, the time when it's really rotating between here and here, that goes very fast, where it's on the microsecond scale, whereas this is on the millisecond scale. And so since we're only acquiring our data at 1,000 frames a second, we're mostly collecting the information concerning where it is during the catalytic dwell. So instead of getting one sinusoidal curve, we're now getting 3 sinusoidal curves, and you can see the 3 right here, 1, 2, and 3. And notice, you may say, well, wait a minute, they're not exactly evenly offset by 120 degrees. No, that's right, because we can't control exactly the orientation of the nanorod. So the fact that we see this tells us we're looking at F1, and it also gives us an angular dependence of that particular molecule. So we can see in 3-dimensional space what we're looking at. Okay, so we've also modified the microscope where we split the beam through a beam splitter. It here comes through the pinhole. We split them, and now we go into 2 photon counters with 2 polarizers. These polarizers are mounted so that we can precisely rotate them. And so we rotate one versus the other by a few degrees. And now, when it's rotating counterclockwise, we can tell versus clockwise, because when it's counterclockwise, then one of the sinusoidal curves will go before the other one, and that will be reversed. And of course, we got consistent results that showed that the F1 rotates counterclockwise. Again, here we're collecting data at relatively slow frame rates, so we're not getting an awful lot of information here in the actual rotation. It's mostly determining how long the dwells are here. But of course, that information of what is it doing when it's really rotating, that's the most interesting part of the whole thing. So with this, we've actually been able to capture the data at rates as high as 400 kilohertz. That's equivalent to about 400,000 frames a second. So this gives us that 400 gives us 2.5 microsecond resolution. Most of our data we've collected at 200,000 frames a second, which is 5 microsecond resolution. And that gives us a fair amount of resolution to capture mostly what's going on. So what we do is at the very beginning of the experiment, we identify our rotating nanorod and we line the pinhole up. Before we start collecting data, we rotate the polarizer so it's aligned with, that the minimum is aligned with one of the 3 catalytic dwells. Consequently, when it's rotating, one of the 3 subsequent power strokes will start at a minimum, go through a maximum, and then to the next catalytic dwell, and then on this to the third, and then finally this will repeat so that it now comes up and back down here. So what we realized is that when we can take these long data sets that have several thousand power strokes, and we can write an algorithm where we start at a minimum and we know we go through a maximum, then that means that it's rotated 90 degrees. So we just collect the power strokes from this subset of data. At first we were also collecting the third one, but we found that we get so much data from this compared to what was possible with fluorophores that we've just concentrated on this first power stroke. And we call this a transition. So here's some real data. Here's the catalytic dwell going along on it in its millisecond time scale. The power stroke begins and the algorithm pulls this out. You can see here's the orientation of the nanorod and the light intensity. It comes up through the maximum and it keeps going until it gets to 120 degrees and goes back to the next catalytic dwell. So we have the algorithm pull this power stroke out. Then we use the algorithm to use an arcsine square root function to convert from photons per microsecond as a function of time to rotational position as a function of time. And the first 90 degrees uses this, but now it's going in the opposite direction. So we have to use a different equation to get from 90 to 120. We take thousands of these, convert these, we average them, and we then take the derivative, and that way we can see the difference between each of these data points. And that way we get the velocity. And so here's the average velocity plot. And you can see that after this is the end of the catalytic dwell, and it starts off, it's going pretty fast, but then it slows down, speeds up, slows down, and then we've divided this into 2 phases, divided over 60 degrees. I will say that in the early days, people felt that the velocity would have to be constant throughout the power stroke. Without any data to support that, with the early measurements that suggested that the efficiency of the F1 motor is 100%, which would defy the second law of thermodynamics. Now it is very high, and there are conditions that were used in those experiments that conclude 100% that are fairly convincing. But when the motor is running on its own, it's not slowed down by drag imposed on it by a huge probe to see the rotation, it's definitely not going at the same speed throughout the power stroke. So the next thing that we did was to do the first experiment was done at saturating ATP, and that's done at 1 millimolar ATP. But when you start to limit the amount of ATP, then it becomes rate limiting to the overall rate. And what we find is that that actually changes, especially right in this region, the velocity of the rotation. This is due to the formation of what is known as an ATP binding dwell. And we found, looking at the dwells where rotation has completely stopped, it's waiting for ATP to get there because there's just not enough ATP in solution. So it can't get there in time. So it sits there and waits for it. And we found that it is dependent upon the rotational position at which this occurs. Now, there was a battle at the time of ideas of how this worked, how the motor worked. And it was thought that one of the strains of thought was that in the empty catalytic site, the lever arm is open, and that as the ATP would bind, that would close the lever arm and push against the gamma subunit as it would against the camshaft to rotate it. So we addressed that by comparing what we saw, the rate that we saw using ATP, the normal substrate, with ITP. And the Allen Senior Lab had determined what the dissociation constants were of ATP versus ITP. You can see ITP, because the base is different, it binds much more loosely than does ATP. And sure enough, we saw that it went a lot slower with ITP than it did with ATP. So that gave us the indication that, in fact, the binding constant of the ITP was causing the lever arm to close and force the gamma around. Now, this was confirmed by a completely different kind of experiment using molecular dynamics simulation by the Karplus lab. And so Martin Karplus, who had pioneered this molecular dynamics and got the Nobel Prize for it a few years ago, then he had done this experiment where he forced the lever arm to close on the computer, and then he looked at various locations up and down the gamma subunit to see what was the torque generated. And so here, this red spot here corresponds to this line of data across here, the blue, and so forth down here. And then this is rotational position as it's going around. Here's the end of the catalytic dwell, ATP binding dwell. And so when you have red, that's where it's generating the most torque over that period of rotational positions. And when you convert these data into something that's equivalent to what we were seeing, it fit quite well from the point where ATP would bind, the blue data are his simulations, to what we were actually seeing, which confirms that when ATP binds, that lever arm closing is pushing the gamma subunit around, at least for phase 2. So it's also been noted that ADP inhibits the F1 ATPase rotation. So in this experiment, we compared what we were seeing at saturating ATP, and under these conditions, it's still 1 millimolar magnesium ATP, because that's what really binds the substrate. And we compared that, and we looked on a 3D plot, which is difficult to interpret. So I've sort of summarized the results. I'll say that in a minute. And so here, this is at 0, and then this is the dwell duration, and in the vertical is how often the dwells occur. So you can see here, it's easy to sort of see that this comes down and is scooped down as far as dwells go, et cetera. But when you add ADP in high enough concentrations to inhibit, we see that now this has come up and that this is extended much more. So this was seeing an ADP inhibition that occurs right around here at about the same place as where ATP occurs. That's not new. And so this is the plot we get of the increased dwells we get from ADP inhibition. But we also saw a difference over here as well, that the velocity was actually slowed down. It was slowed down to the greatest by the presence of ADP right through this region, but it extended over this period. And so we concluded that the ADP binding here was competing with ATP for the binding to the empty site, whereas this effect of ADP, the high concentrations of ADP, were preventing the dissociation of ADP from the beta DP site, a separate site, which had not been appreciated previously. So here's a summary of these first results. Here, at rate limiting ATP, where we'll see an ATP binding dwell, we'll start off at the catalytic dwell, and that's where ATP gets hydrolyzed here. Then with phosphate release up at the beta empty site, once that happens, that starts the rotation. At limiting ATP, it has to sit around waiting for ATP to come in. That closes this lever arm and allows the power stroke to continue. Now you go into a catalytic dwell where it's this site that hydrolyzes the ATP. Okay, so what happens at high ADP, where we have lots of ADP lurking around, waiting to bind? Then we start off the same. Catalytic dwell here, ATP gets hydrolyzed. But now, when the phosphate comes off and it starts to rotate, the ADP outcompetes the ATP, gets in there and won't let the ATP bind. Finally, the ATP can dislodge it, and now it starts to rotate again. But this last bit of rotation is slowed down by the slow release, and all these ADPs slow down this release and slow down the rotation through this period. So at this point, up until now, I've talked about the E. coli ATP, F1 ATPase. We've also done this with the thermophilic ATPase, and we've extended this to other ATPases too. There is a larger superfamily of these, of which here's E. coli, what it looks like. The spinach chloroplast looks very similar. The bovine has quite a different peripheral stalk. In fact, this peripheral stalk can make dimers attached by the peripheral stalk. And then there is the V-type ATPase that is found in all eukaryotes. This is from yeast. And you can see it has not just one peripheral stalk with the subunit A attached, but it has 3 peripheral stalks. This will only go in the ATP hydrolysis direction. And then there are this hybrid kind. They're bacterial, but they're archibacteria. And so they're oftentimes called the A-type ATPase, but some groups call them the V-type. Because they have multiple stalks. They only have 2 stalks. And they also differ in the construction of the central stalk, the gamma subunit. You can also see the size of the C ring changes too. For the peripheral, the F1 or V-type, VA type, then we'd see the same kind of profile for accelerations and decelerations. Admittedly, it's slower through here than it is for E. coli, and that's consistent with the measurements of torque, that the thermophilic and the VA type have lower torque. One major difference that we found is that when we go to limiting ATP, then we don't see an ATP binding dwell that occurs right in here. We don't know why that is. And another lab has also looked at the bovine as well as human, and they find that in this group, there is an additional phosphate dissociation dwell that occurs right out here. Again, we have no idea why there is a difference. What's the basis for those differences? So up until this point, I've been saying that mostly the ATP binding dwell occurs at one position. Even though I showed you that it occurs over this range. The lab that first showed this was Yasuda, published in Nature. they got these distributions where this is the distribution of the location of the catalytic dwell and the distribution of the ATP binding dwell, which they said was about 40 degrees away. And because of the low accuracy in determining rotational position, they assumed that this was just the error in their measurement. So they came up with this ATP arrives in time hypothesis that I already mentioned. And that is, you start at the catalytic dwell, and when you're at a saturating ATP, the ATP gets there before it reaches 40 degrees. So it just keeps rotating, no ATP binding dwell. But over here at limiting, it gets to the 40 degrees and then has to wait for ATP to come. But what we found is it more complicated than that. That here's what it looks like when you have saturating. We don't see any ATP binding dwell. This is the rotational position versus time, and these are the photons, this is the actual raw data. So no ATP binding dwell. But when we're at limiting, now we see ATP binding dwells interrupting the rotation. Notice that it doesn't oscillate a whole lot during this time, it's just stuck. But they can occur early, and they can continue all the way up to late, so to the point where it's like pretty close to 50 degrees. And so when we superimpose how often these occur on top of the data of the earlier Japanese work, we see that it overlaps quite well. So that tells us that while the catalytic dwells always occur in the same location, these ATP binding dwells can occur at relatively different locations over this entire phase. And our other work that we have yet to publish shows that this makes a real difference in how the power stroke, the rotation, goes during that time as the location of when the ATP binds. So now we've done temperature dependence of the angular velocity curves. And we analyzed the data by Arrhenius analyses to get energy terms of how much energy is involved in consuming as it rotates. So in order to do this, we collected data over a temperature range starting at as low as 16.3 and is going as high as 44.6 degrees. And we found that we could collect the data at this, and we could start at a low temperature with the same molecule, go all the way up to the high temperature, collect data, and we could go all the way back down again to the low temperature. And when we collected the data at the low temperature before and after going to high, we still got the same result, saying that the high temperature, the excursion to high temperature had not damaged the protein in any way. A process known as hysteresis. You want to avoid that because you can't determine what, you're not getting a consistent answer in your result. You're mostly looking at the damage to the protein, so no damage. Anyway, you can see that there were differences, although it still followed the same general pattern, there were differences. So now for each different rotational position, we could construct an Arrhenius plot. And of course, in introductory biochemistry, it's typically taught that when the temperature goes up, the rate of an enzymatic reaction will double for every 10 degrees increase. And so certainly up here during phase 2, we did see that occur. Now, to do an Arrhenius plot, you plot it as 1 over temperature. So the high temperatures are here, low temperatures are here. And we do it as a log of the plot. But you can see up here, 85 degrees, it's getting faster as it gets hotter. And that is same for 76 degrees. But notice what happens once we get down to phase 1. It turns around and goes the other way. Now it actually goes slower. And at first we thought maybe we've made a mistake here. But no, this is actually telling us something really important about the protein. And what it's telling us is that phase 1 rotation uses energy from elastic energy within the protein to get it to rotate that far. This is commonly what you see with rubber, of how rubber will contract when it's stretched and then allowed to relax as a function of temperature. So here's splitting up our Arrhenius plots every so many degrees. You can see how it goes up, gets steepest right in here. But now as it comes down to going from phase 2 back to phase 1, here we don't even see any change. And now it's going negative and then back up to 0 at the next catalytic dwell. So from this, then we can plot the activation energy. You get that from the slopes of those previous plots. And here the activation energy is negative. And in fact, it forms an inverted parabola. And so if this is like a rubber band airplane, so right here, that you would use energy from this twisting of that rubber band or the gamma subunit, then we can calculate the spring constant, the amount of energy derived by twisting it by this equation, and we get a spring constant for the gamma subunit of about 50 kBt per radian squared. It has a maximum of about 34 degrees. We also saw another a parabola-shaped decrease, and that we assumed was due to a spring constant that was stiffer with 150 kBt per radian squared. So now what we then looked at was, well, what happens, where do the ATP binding dwells occur? It turns out I could take the same parabola here, superimpose it on the probability of the ATP binding dwell. So ATP binds most commonly right here. And so you can imagine that here you have at the catalytic dwell, tightly wound spring, it starts to rotate, but now it gets down to the point where it's at its energy minimum. This is where ATP binds and now propels it, closes the lever arm to drive the rest of the rotation. What's going on here? Turns out this is where ADP binds and causes the dwells. And you can see here, that this would superimpose on here. This is what gets slowed down by ADP inhibition by binding at the beta DP site. And I can superimpose that on top of this curve as well. And that's probably not by accident, because this is its low energy point. And so this is where ADP would be most likely to be most effective in inhibiting the rotation. We can take the activation energy, convert that to enthalpy. It's essentially multiplying by a constant, so you get the same pattern. But with that, then you can derive both the free energy of activation and the entropy. You can see it's mostly an entropy-driven process. And the free energy then of activation fits very well with those measurements that were made by other labs for the catalytic dwell. Here's before ATP is hydrolyzed, here's afterwards, and that's where we get our free energy of activation. And then it ends back up here at a comparable location. And notice that this is taking the velocity curve and flipping it upside down, which makes sense that you would have where it's going the slowest here, that's where you have the highest free energy of activation, and conversely, here where it's going the fastest. So we knew how much energy we were putting into the system because we had controlled the concentration of phosphate, ADP, and ATP when we did this. And you can calculate what's the ATP energy that we are providing it, divide that by this free energy of activation, and now we get our efficiency. It's not 100%, it's still very high, but it's not 100%. We haven't broken the second law of thermodynamics. At this point, we've also started to look at what happens during the catalytic dwell. Here's the power stroke, comes up 120 degrees and goes into the catalytic dwell. You can see it doesn't just flat, but it's oscillating. So it's kind of wiggling back and forth like this. Again, that's energy from elasticity. And so there's a lot more to say about that, but based on that, we can now put together a sequential mechanism where during the catalytic dwell, we're seeing oscillation. ATP hydrolysis changes that oscillation of the gamma subunit due to twisting. Then you get phosphate release that initiates the counterclockwise rotation of the gamma subunit in the phase 1. ATP binds, closes the lever, and now it finishes off the power stroke to go back to the next catalytic dwell. Now, in order to make this work, in order to store energy, if this really is the gamma subunit being twisted, you've got to have something that tethers it down here. Because again, the good analogy is this rubber band airplane. In order to store the energy so that once after you twist it, you let go of the propeller and it starts to spin, that the way it's stored is you have to have an anchor here. We have found that there is an anchor. The empty catalytic site specifically makes very strong electrostatic interactions to a point on the gamma subunit. We've mutated these residues and we find they dramatically decrease the amount of ATPase activity measured in ensemble experiments. And we've now collected data on single molecule experiments using these same mutants, and they have very interesting results that we're in the process of publishing now. So this all suggests that the point going from phase 1 to phase 2 is probably the exchange of the catch loop from what was the beta empty site to what will become the next beta empty site as it rotates. And again, coming back to the work of Karplus that he did just recently, where he did these energy calculations, he finds something very similar, phase 1 to phase 2, and this crossover point right here, we project that our data will be consistent with the exchange of the catch loop as it goes from one to the next site. Okay, moving on to the FO motor, then all of these different complexes for the F1, they work in 3 successive 120-degree steps, each separated by a catalytic dwell. So to get a full rotation of 360 degrees. However, there is a huge difference in the number of C subunits, and each C subunit has a carboxyl group that allows the binding of 1 proton at a time. So for the bovine, which only has 8 C subunits, each proton causes a 45-degree rotational step. So you'll have 8 times 40 degrees to get to full rotation. E. coli has 10, 36 degrees for 360, and the cyanobacterial one has 15. So you only get 24 degrees for each step. We've been examining the E. coli one, has a C10 ring, so we're looking for 36 degree stepping in this process. Does it rotate? Yes, it does. So anyway, this is what we're going to be looking at, is this interaction down here. Several people had been trying to see something going on as far as stepping in the FO motor prior to our work, but they had been unsuccessful. But I'll explain why that is in a minute. So we now, with the advent of cryo-EM, we can get structural information of the FO for the first time. And here is the E. coli FO motor and the C10 ring surrounded by subunit A. And you can see the helices come down at an oblique angle through the membrane with these groups that have been believed to be important for having the protons come in and feed the C ring with a proton, and also to take the protons off. So here is coming in, thought I had a little input arrow here. So the protons come in here, protonate here, that moves this out. Now this is exposed to the membrane, so you can't have a charged group of a carboxyl group here. So the protonation neutralizes the charge. It sequesters the carboxyl group until it makes its way around here with successive protonation steps. And every time there's a protonation step, there has to be a deprotonation step as it comes off over here. These two half channels then are separated by R210, this very strongly positive charged group that's in the membrane and that separates the two halves of the channel. So how we did this, in order to make this work, is that we purified the F1FO using detergent, as other people had, but instead of using the detergent solubilized, which they could never see stepping, because in the presence of detergent, the subunit A dissociates just a bit from the C ring. But to keep them happy, we embed those in a small nanoscale lipid bilayer that's inside a nanodisk. We're one of the first people to use nanodisks. And they're just amazing because it's a very good surrogate for a biological membrane, and yet it's on a nanoscale where we can do single molecule experiments. The rest of the setup is the same. Notice that if you are just using detergent solubilized, this is your ATPase activity. Within 4 hours, it's dead. But if you put them in nanodisks, you're still 100% at 8 hours. In fact, we've gone as far long as 2 weeks. You only lose about 20% after 2 weeks. Of course, we don't do that. We just do it for the time we're collecting data. But it makes a huge difference in how well FO functions. Here's the raw data of counterclockwise rotation in degrees versus time when you just have F1 and it's saturating ATP. We still just drive the rotation with ATP for this F1FO. But when we have F1FO, we don't just get continuous rotation. We get these, what we call transient dwells, where it stops rotating, stops rotating, stops rotating, and it turns out, averaging over thousands of these, the transient dwells occur about every 36 degrees. In other words, for each C subunit as it rotates past subunit A. Here's another list showing all the different kinds. In fact, what we see is that it's not only stopping rotation, but in many cases, it actually, while this is counterclockwise rotation, it can not only stop the ATPase-driven counterclockwise rotation, but it can force it to rotate in the clockwise or ATP synthase direction. And it can do so by as much as 1 C subunit. Now, over the average, here's sort of an example. This one went 10 degrees clockwise. This one went 25 degrees clockwise. 70-some% are actually can grab, subunit A grabs the C ring and pushes it in the synthesis direction. 28% that only grabs it, stops it. The average, it's not really the average, it's the most common number of degrees that can push it clockwise is 11, with the maximum about 36 degrees. So what we're really looking at here, remember, it's still dominated by ATP hydrolysis, but it's amazing. This is the little motor that could, because it can stop that ATPase and drive it in the synthesis direction. So first of all, if we just look here, these sometimes, even though we have F1FO, it still looks like just F1 is there. And basically, you see it rotates like F1. But here, where you see it, it just stops it. It stops, stops, stops. Here, where it goes clockwise, it goes this way, back, and back each time. That's what we're actually looking at. So we collect a series of data. In one data set, we'll collect about 300 of these power strokes. And then we take the average of what percent were having transient dwells. Now we take thousands of data sets from 51 different molecules on average, and now we get a distribution. And you can see that even though the average of this distribution would be down around 30%, some of these molecules are pumping out 80% having the transient dwells. So we've done this as a function of pH, and we find that at high pHs, we still see over 20% are still having transient dwells, and that this increases up to an average of just about 50% down around 5. And it's noteworthy that we chose between 5 and 9, because when you set up a pH gradient across the membrane to drive ATP synthesis, you typically use a delta pH of 3, and you go as low as 5 and as high as 9. So this is under conditions that would be consistent with what we're seeing there. Now the blue curve is a Henderson-Hasselbalch curve, but you can see it doesn't fit all that well. That's because there's many more than just one protonatable groups that's responsible for this dependence. Over the pH range, you can see it stays 36 degrees, doesn't really change much. The duration of the dwell does change a little bit, but it goes from about 120 microseconds up to as long as 150 microseconds. Now, the one thing to note is that this is comparable to the time, the duration that is observed during ATP synthesis. So again, we're seeing something that we would see during ATP synthesis. So here's the distributions that we see for the wild type as a function of pH. You can see it starts low, high pH, and it goes up higher as we go down to pH 5. You can see here's where we have 100% of some molecules that every single power stroke has transient dwells. What confused us for a long time is that this didn't fit to a nice simple Gaussian. In fact, in order to make it fit, we had to fit 3 different Gaussians to it. But then when we thought about this, we thought, gee, the gammas and epsilon subunits are asymmetric so that it's in 3 different positions. And you can see that by cryo-EM structures where you're actually looking, you can see all 3 different positions. And because we have 10 C subunits, but only 3 catalytic sites, then during one of the 3, we're going to have an alignment. But in the other ones, we're going to have, it's not going to be aligned. The F1 is going to be out by a good 12 degrees, and it's going to be out in the opposite direction over here. So we hypothesize that the twisting that we're seeing is going to, of the gamma subunit, is going to help it in some cases, and it's going to hinder it in other cases. So we take these distributions and we plot those, and we go, still we see pH dependence. And now we have a low probability, medium, and high, 3 bears, and that fits with this. So once these structures came out, then we hypothesized, looking carefully at the 3 structures. Remember that for each 120 degree steps, you got 10. So we're going to have 2 3-step, 1, 2, 3, 1, 2, 3, but then this one's going to be 1, 2, 3, 4. So when we looked at the 3 different structures, then we hypothesized that one of these structures, with the alignment that we have of gamma and epsilon, will give rise to the high probability, and the other ones will to the others. We're collecting 50 molecules to get our distribution. We don't know which molecule would align which way. The nanorods attached to the gamma subunit, and we can't tell which it is. But we can see that from the high, low, and medium probability. Okay, so now looking at the extent of the back step, it didn't really look all that interesting at first as a function of pH. The biggest thing we saw was that this group that didn't have a back step, that did change as a function of pH. But we found that when we took a subtraction of this, that the pH 5.5 and 5, they were the lowest. So we took the different spectrum of 5.5 and we subtracted each one of these, the 5.5 from each one of these. And what we found is that there was a difference underlying this, and that it fit to a Gaussian that had a maximum of about 11 degrees. So we were seeing a change as a function of pH of 11 degree steps primarily. We didn't know what to make of that. However, in just the last few weeks, A new, much higher resolution structure of the E. coli cryo-EM by cryo-EM has been revealed. I just made this up last night because it's really quite exciting and it confirms everything that we've been working on with our single molecule transient well results. They find that, if I go back, that where we had predicted we would see the high transient dwell probability step, that structure is consistent with, in their high resolution, that they not only get one structure for the FO, but they get two. And the only difference is the rotational position of the C ring. That here is the one structure relative, here's R210, and there is the other structure. It differs, they claimed it differed by 36 degrees, but it's really not, it's 25 degrees between the two conformations of the C ring. Well, 36 minus 25 turns out to be 11. And so we are seeing up to 36 degrees, but we're also seeing these 11-degree steps most often. Where could that come from? Well, one would be a proton-dependent 11-degree step, so we'd get a total of 36. And here's one where we see 11 degrees. The other way is that we see the proton-dependent donation here, and that causes the movement of 11, so 11 plus 25. We don't know which one this is yet, but we're working on that. Anyway, it was a nice confirmation of the last few years of our work. And so with that, I'll end. I want to thank everybody who has really stood at the bench and at the microscopes for hours and hours doing all this work. At this point, it's now a fairly large group of people. They all weren't in the lab at once, thanks for support from a wide variety of funding sources that has enabled this work to take place. So thank you very much, Nina.